months. Reproduction, an important process in human beings, involves the fusion of the male and female gametes called sperm and ovum. These gametes are formed by a process called gametogenesis. In males, the process of gametogenesis occurs in the testes and is called spermatogenesis, whereas in females, gametogenesis is called oogenesis and occurs inside the ovary. Interestingly, spermatogenesis and oogenesis begin at different ages. Spermatogenesis begins at puberty, usually between 12 and 13 years of age, and is divided into two stages: spermatocytogenesis and spermiogenesis. Spermatocytogenesis, the first stage, occurs in the seminiferous tubules, whose inner walls are lined with spermatogonia, the diploid male germ cells containing 46 chromosomes. Some of these spermatogonia divide mitotically to form two daughter cells. Of these two daughter cells, one remains the spermatogonium, while the other one becomes the primary spermatocyte. This primary spermatocyte divides meiotically to form two equal cells called secondary spermatocytes. Unlike primary spermatocytes, secondary spermatocytes are haploid cells containing 23 chromosomes each. Spermatocytogenesis is succeeded by spermiogenesis, during which the spermatids differentiate into an elongated and tadpole-like spermatozoa or sperms. However, during this process, the heads of these sperms remain embedded in the Sertoli cells, which provide these developing sperms with nutrition. Post-spermiogenesis, non-motile sperms are released into the lumen of the seminiferous tubules by a process called spermiation. The lumen has testicular fluid that helps transport the non-motile sperm to the epididymis, where they mature and become motile. During ejaculation, sperm from the epididymis are transported via the vas deferens to the urethra in the penis. As the sperms pass through the different tubes, it mixes with fluids secreted by the seminal vesicles and prostate gland to form a mixture called semen, which is ejaculated by the penis. Did you know that semen released during a single ejaculation contains 200 to 300 million sperms? Microscopic studies have revealed that a typical sperm is covered by a plasma membrane and has 4 distinct parts: head, neck, a middle piece, and a tail. Inside the head lies an elongated haploid nucleus containing chromosomes. The anterior part of this nucleus is surrounded by acrosome, a cap-like structure filled with enzymes that help fertilize the ovum. The sperm's head is followed by its neck and middle piece, which is filled with mitochondria. These mitochondria break down glucose in the semen to produce ATP, an energy molecule that powers the movement of the sperm's tail. The formation of sperm is triggered when the hypothalamus secretes high levels of gonadotropin-releasing hormone, or GnRH. This GnRH acts on the anterior pituitary gland and stimulates the secretion of the luteinizing hormone or LH and follicle-stimulating hormone or FSH. LH in turn stimulates the Leydig cells to synthesize and secrete androgen, a hormone that triggers spermatogenesis. FSH, on the other hand, acts on the Sertoli cells to stimulate the secretion of substances that nurture sperm cells during spermatogenesis. While spermatogenesis occurs at puberty, oogenesis is triggered before a woman's birth. When the germinal epithelium cells in the ovary of the fetus divide mitotically to form gamete mother cells called oogonia. A few oogonia divide again to form two immature cells called primary oocytes. The primary oocyte then gets enveloped by a layer of squamous granulosa cells, and together they make up the primordial follicle. The oocyte further develops as the fetus develops, but the granulosa cells do not change till puberty. In fact, primary oocytes enter the prophase of meiotic division and remain suspended till puberty. Did you know that at birth, a female has approximately 7 lakh oocytes? Incidentally, no oocytes are formed after birth. But by puberty, most of the primary oocytes degenerate, leaving only about 4 lakh in the ovary. At puberty, the release of LH and FSH hormones stimulate some primary oocytes to resume meiotic division that had begun during the woman's fetal stage. Moreover, due to FSH, the squamous cells of the primordial follicle enlarge and become cuboidal. The dividing primary oocyte and the cuboidal granulosa cells now make up the primary follicle. The primary follicle is transformed into a secondary follicle when it enlarges and is surrounded by several layers of granulosa cells, which in turn are enveloped by a developing theca or capsule. Later, these granulosa cells secrete estrogen, which creates the antrum, a fluid-filled cavity among the granulosa cells. Moreover, the theca becomes evident and gets organized into 2 layers, theca interna and externa. This structure is now called the tertiary follicle. As the secondary follicle transforms itself into a tertiary follicle, the primary oocyte within the follicle completes meiotic division, which is unequal and yields a large haploid secondary oocyte and a tiny first polar body. The polar body is believed to degenerate, leaving the tertiary follicle with only the secondary oocyte or ovum. As the tertiary follicle develops into a mature follicle or Graafian follicle, the antrum grows and the secondary oocyte and surrounding granulosa cells are pushed towards one end of the follicle. The granulosa cells secrete a glycoprotein substance that forms a membrane, the zona pellucida, around the secondary oocyte. The secondary oocyte or ovum is released from the Graafian follicle when it ruptures due to a surge in LH in the body. This process, called ovulation, causes the oocyte or ovum to be placed in the oviduct, where it awaits fertilization by sperm. In this way, the ovum and sperm are formed through a long and intricate process called gametogenesis.